So first thing we need to do when we come back into the lab is sort our sample. I use different size mesh to make sure that we see all the, we don't just pick the big things, but we pick the small things as well. So I use a 5 mesh or 4,000 microns on top of a 10 mesh, which is 2,000 microns, on top of a 35 mesh, 500 microns. So put the smallest on the bottom, largest holes on the top. And usually when I come back from the field, I have the samples in a little plastic baggie, but right now I have it in a bucket. We just pour things through. I use RO water to rinse everything because we don't want the chlorine to hurt the organisms in the regular tap water. So I use reverse osmosis water. And I just rinse everything through the sieves. I actually messed up filming this the first time, but usually when it's fresh from the field, you'll see the water is very brown colored, and that's a lot because part of it is just particles in the water like sand and fine particles, but some of it is actually diatoms, so it's kind of an orangey brown color, and we usually want to get rid of that. So I take each sieve size, put it in take each sieve size and put it in a different bin. And I usually want to make sure that this doesn't still have any organisms in it. You can pour it through the smallest size again. I saw a few organisms that got free because it was kind of low. Doesn't hurt to get rid of some of the water and some of the debris. And then I rinse the sieve off into the bin. Checking to make sure that no organisms got free. I do the same for all of the bins. I'm going to keep the organisms in water as much as possible. Try not to let them sit out for too long. Just check to make sure no organisms are still stuck on the sieve. They do like to stick. Sometimes it's just leaves or other gunk. It's not always organisms, so you can look to see if it's stuff is moving around. If it looks like it's an organism, you want to make sure you get it all off. Off the best we can. Things like to cling. now we have the larger sizes. Looks like there's some larger things in there. A lot of leaf material as well. Here's the medium size. You can see a lot of little organisms moving around in there. And here's the small size. See, lots of little things. Now, most of these organisms like to live under rocks, so they will immediately try to hide under each other, under other debris, whatever. So sometimes you have to kind of stir things up so you can see them out of hiding. So after we use the sieves to sort things into these three size classes, then we take these bins and pull out the organisms one at a time and sort them into different classes. Some of the tools used for sorting the organisms include things like transfer pipettes, which have different sizes. This is a regular one with not cut, and then cut off the tips to make different sizes. I've taken a dissecting needle and hot glued some mesh on there, or spoon, or some soft touch forceps. And then we use them to suck up the different organisms and place them in pipettes, petri dishes I mean. And we try to stick things that are the same type. So this one, these two are the same type, so they would go together. But this is a different type of organism. So you can see they look pretty different. So we would put it in its own dish, and like I said, you can use here. You can use different tools to capture things. Smaller things are things that move fast. It's nice to use a pipette to suck them up. You can also, for the slow things, you can often just grab them with the soft touch forceps. And the idea is we just pull things out and sort them by type, and then eventually we put them in a vial with some 70% ethanol and a little label printed out on waterproof paper. Repulled? Little waterproof label that I stick inside the vial. It's printed on a laser printer on waterproof paper, and that will stay on even in 70% ethanol. And then we label the vial with what microhabitat it came from and what part of the river, upstream or downstream, and what day it was sampled and all those kinds of things. And then later we come back and count and identify everything, because you really need a microscope to identify these organisms. So we need to take our time identifying things with the microscope, and we'll do that. It takes a lot of hours to identify and count everything, so we preserve them and identify them, taking a much longer time to get them identified. So that's the general process. When we're all done sorting, we place them in the organisms in vials by type with a little label in there, and I labeled the top with tape as well, different color tape, but I don't have it on here. We preserve them in 70% ethanol. Right now these are in plain water because I'm actually returning them to the river. So we store all the organisms in little vials until we can have time to fully identify them. Here's an example of a bunch of vials. So they're all labeled with the microhabitat they came from, and each vial gets its own number, and they all have organisms and tags, and they're preserved in 70% ethanol so that we can take our time identifying them. It takes a long time to count and identify all the organisms.